Bone is the predominant tissue of the skeletal system. When we examine it, while there is an abundant extracellular matrix, there also are cells. There are 4 different types of cells in bone tissue, but the stem cells, osteoprogenitor cells, are not discussed here. They turn into osteoblasts, which build bone tissue initially and then modify it later on, repairing and remodeling it as necessary. They lay down the extracellular matrix of bone by producing collagen fibers and proteoglycans. They then initiate calcification by secreting calcium and phosphate ions to form a crystal called hydroxyapatite. This is also what's found in the dentin and enamel of teeth and is used in dentistry and found elsewhere in nature, like the shells of bird eggs. It's a very hard, resilient crystal, and it's what gives bone its supportive capability. When osteoblasts secrete as much matrix as they can, they turn into mature osteocytes. These cells work to maintain the bone matrix and keep the tissue healthy on a day-to-day basis. So they're surrounded by that lacuna, connected through those canaliculi, and they respond to day-to-day changes, making sure that bone tissue remains healthy. Osteocytes comprise most bone cells, the vast majority, almost all, 90 to 95%. Osteoclasts come from the white blood cell line, so they're not related, but they have that osteo in there because they're functionally bone cells. They are megacells comprised of 50 or more white blood cells that come together, and on one edge of the cell, called a ruffled border, which faces bone tissue, they will secrete chemicals that break down existing bone tissue, a process called reabsorption. You might think this is a bad thing, but this reabsorption is necessary for the initial creation of bone, as well as repair and remodeling. Osteoclasts also will function in blood calcium homeostasis by breaking down existing bone tissue to release the calcium ions it contains. To remember these bone cells, think osteoblast B for baby builder cell. These are immature cells that build tissue. So blast, baby builder. When they grow up, they turn into the cyte, osteocyte, mature maintenance cell. Just like when we're babies, we don't have a house of our own, and then as we grow, we can create our own house. Osteoclast, think carve or crusher cell. These cells carve out or crush bone tissue. Here we can see, zoom in here. So here we're looking at the osteons in our spongy bone, and then when we zoom in, we see just one osteon here. And the cells that live within those lacunae we talked about earlier are the osteocytes, surrounded by that mineralized matrix, housed within the lacunae, and connected to each other via processes in the canaliculi. Over here, we'll see bone creation cells. So as part of the inner layer of the periosteum, also found in the endosteum, are osteoblasts that work to build up bone tissue. We'll also find osteoclasts here. And you can see the osteoclast looks quite different. It's multinucleate because many cells come together to form it. And then on one side, there's this special ruffled border from which the cell can secrete enzymes that break down existing bone tissue. And these are found along with the osteoblasts in periosteum, endosteum, and lining the central canal of osteons as well. Now that we know our bone cells, we can talk about how bone is formed through ossification, which is the biological term for bone formation. This process begins before we're even born in utero, and both osteoblasts and osteoclasts play a role. Of course, osteoblasts are very important because they're the cells that build up bone tissue. And as we just talked about, it will build up as much as it can until it's completely surrounded, and then it's termed an osteocyte. It's grown up. Osteoclasts also play a role, which is less intuitive, but they need to break down existing tissues so osteoblasts can lay down bone to replace it. And they also work to remodel the bone as it's forming. There are two types of ossification or bone formation. These two processes share some characteristics. They both build both types of bone tissue. So, spongy, then compact bone result from both processes. Also, there's going to be some kind of pre-existing tissue that ossification replaces with bone. So, both processes are going to start with something instead of creating bone out of nowhere. The two processes are called intramembranous ossification, which is creation of bone within a membrane-like sheet of tissue, so in a membrane, intramembranous. And endochondrol ossification. Endo for within, chondra, like a chondroblast, chondrocyte, indicates cartilage. So within a cartilage model. Intramembranous ossification is the simpler of the two. This is bone formation within an embryonic connective tissue sheet or membrane-like structure. This is relatively limited, only forming the skull bones, little bit of the facial bones, and the medial bit of the clavicles. So basically think the skull. Head up, neck up would be intramembranous ossification. Here, we're going to see osteoblasts aggregating or lining up on the surface of that connective tissue membrane, then depositing bone matrix from multiple, 2 or more ossification centers to form the trabeculae of spongy bone. Then the trabeculae from each ossification center will grow out or radiate out and fuse with the trabeculae created from other ossification centers. After we have fusion, we'll see that spongy bone remodeled, enlarged, or replaced by compact bone tissue on the surface. These are the general steps, and they coincide with the image from your book, which is very exciting because earlier editions did not have this image and it was quite difficult to understand it. But now we have a nice step-by-step guide. So number 1, first step, we'll see osteoblasts within those multiple ossification centers, laying down collagen fibers and mineralizing those fibers on the surface of the connective tissue membrane. This forms the trabeculae of spongy bone. The osteoblasts become osteocytes once they secrete as much matrix as possible. So this is step 1, where we have that connective tissue membrane in the background. Then we see these osteoblasts lining up to secrete bone tissue. And this process has already started because we see some of those osteoblasts have already become completely encased in that bone tissue. So they are osteocytes within lacunae connected by canaliculi. Step 2, more osteoblasts on the surfaces of the trabeculae do what they do best. Lay down more bone tissue to build up trabeculae, and those trabeculae just grow and grow, join up with the trabeculae from other ossification centers to form spongy bone. So that's what's happening here. We're seeing a single structure formed that is all trabeculae of spongy bone tissue, and this structure continues to grow as the osteoblasts on the surface continue to secrete that bony matrix. Step 3, the cells within the spaces of the trabeculae are going to form that soft red bone marrow tissue, which will fill all the spaces in between those rods and beams of the bone tissue. And surrounding cells are going to form that outer periosteum. The cells within the periosteum, in that inner layer, are going to secrete compact bone tissue. So the surface of these bones will be composed of that stronger, protective, compact bone. Remember, with all bones, compact bone is found on the exterior. So that's step 3 here. We have these osteoblasts on the surface, exterior surface, secreting the periosteum, creating the periosteum, and then osteoblasts that are on these internal surfaces, the trabeculae, will create that marrow tissue, which is not pictured here, but you can see it in the image. This image shows these bones formed by intramembranous ossification, and then also the bones of the limbs and the torso that are formed by endochondrol ossification, which we'll talk about in a moment. And note the fontanelles, or soft spots. The skull bones don't fuse until after we're born. This creates the ability for the skull to compress, allowing for the progress of the skull through the birth canal. And it also allows for rapid growth of the brain after birth. And then about 6 months in, these fontanelles will close up. Endochondrol ossification is more extensive and it's a bit more complicated because there's an additional step. The name hints at this step. This is bone formation within a cartilage model, hyaline cartilage. So there will be a hyaline cartilage model of the bone, which looks pretty much very similar to the bone we'll end up with. And then that model is almost entirely replaced by bone tissue. Here, there's a very strict order to ossification, with a primary ossification center developing first in the diaphysis, which then goes on to form the diaphysis of the long bone. Then 2 secondary ossification centers form later, 1 in each epiphysis. And these then go on to form those epiphyses. Endochondrol ossification forms all the bones, pretty much, besides what's found in the skull. So this includes all of our long bones, all the bones of the limbs, like the femur, the humerus, and the tibia. There are more extensive steps to endochondrol ossification because there is an additional hyaline cartilage model that first has to be created from immature cells before we can even talk about building bone. Step number 1 is chondroblasts building up hyaline cartilage, then becoming mature chondrocytes once they build up as much as they can. After the model is created, the model calcifies or hardens, which kills those chondrocytes as they lose access to nutrients and oxygen. This calcification initiates the formation of a thin collar of bone tissue on the surface of the diaphysis. Once that bone collar forms, a blood vessel from the outside penetrates that bone collar, invading the diaphysis. The bone cells use this entry point to enter the model and start laying down bone tissue. This is possible as osteoclasts break down that calcified cartilage. This gives you the primary ossification center, and this process will continue in 2 directions towards the proximal and distal epiphysis. As this process continues, 2 blood vessels, one in each epiphysis, will penetrate, bringing in bone cells and forming the 2 secondary ossification centers. As this is occurring, ossification is progressing towards each epiphysis, and a medullary cavity is being carved out. So some of that newly laid bone tissue is going to be almost immediately reabsorbed to create this cavity. Finally, when we're done, the original cartilage model is almost completely bone. It's been ossified. The 2 remaining areas of cartilage are the articular cartilage of the joints, where the epiphyses will touch other bones, and the epiphyseal plate between epiphysis and diaphysis, which allows long bones to lengthen after this initial ossification. So we don't have to remain the size of infants. This diagram from your book shows you these steps. So we see the initial formation of that cartilage model by chondroblasts, which then turn into chondrocytes. Then the model calcifies, starting here in the diaphysis. This calcification causes this thin bone collar to form on the outside of the diaphysis. Then a blood vessel penetrates through the collar into the diaphysis, which creates a pathway for bone cells, which create a primary ossification center here in the middle of the diaphysis. Osteoclasts break down the calcified cartilage. Osteoblasts lay down trabeculae of spongy bone tissue. Ossification continues towards each end, towards each epiphysis. And as that is occurring, we'll see a blood vessel penetrate here, just what we see over here, but also the other end. Same thing is occurring over here. And that creates a pathway in for bone cells, which will create a secondary ossification center inside each epiphysis. So, trabeculae of spongy bone are laid down here. And you can see that the medullary cavity forms as initial ossification occurs, and then also modification of the diaphysis at the surface will create that compact bone we need. So by the time we're done, we're going to see only articular cartilage. And if this was an immature bone, we'd see an epiphyseal plate right here that will grow and then be replaced by bone tissue to allow the diaphysis to lengthen. But here we're looking at an adult bone, so lengthening has ceased, and the plate has been replaced by a bony epiphyseal line. So that was initial ossification, the creation of baby's first skeleton. But we're not the same size as when we're born, and the skeletal system has to be modified as we age. Bone growth can occur in 2 ways after initial ossification. Width is going to be relatively straightforward. It's termed appositional growth, because growth is going to occur opposite, allowing the whole diaphysis to get wider. Here, osteoblasts lay down new lamellae on the surface of existing bone tissue in between the surface and the periosteum, and the width of the entire bone is going to increase. This occurs throughout our lifetime. Bone length grows by endochondrol ossification, where the cartilaginous epiphyseal plate that remains after initial ossification is going to grow, then calcify, then be carved out and replaced by bone. So it's really a continuation of this endochondrol ossification that formed the bone, but it's occurring just in this one location of the epiphyseal plate. So until we're about 18, 19, 20, we'll see that epiphyseal plate grow on the epiphysis side. We'll see the number of chondrocytes increase. The chondrocytes then get bigger, and then they die as the cartilage calcifies and hardens. Osteoclasts remove that old calcified cartilage, and it's replaced by bone tissue secreted by the osteoblasts. So the cartilage is growing on the epiphysis side, and then bone is being laid down on the diaphysis side. Eventually, cartilage will cease to be deposited, and the plate will be entirely replaced by bone, forming a line where the plate was called the epiphyseal line. And from then on, we can't get any taller. Our long bones are not going to get any longer. We can see these steps here. So we're looking at this immature bone, zooming in on the epiphysis. And when we do that, we'll see that on the epiphysis side, so over here, we'll see cartilage being produced, and then the chondrocytes will divide, enlarge, die off as the cartilage calcifies, and they kind of stack up like coins here. And then once calcification occurs and the chondrocytes die, osteoclasts will destroy the cartilage, and osteoblasts will replace it with bone on the diaphysis side. So the diaphysis will grow longer and longer. It's a bit clearer to see here. We have just plain old hyaline cartilage here. And then the chondrocytes divide, get bigger, see them stack up. Calcification occurs, cartilage is removed, and osteoblasts lay down bone tissue. And this image shows how we get from immature to mature bone with that cartilage being present in an immature bone and then replaced entirely by bone tissue. And then also our bone being remodeled to form a mature epiphysis lined by articular cartilage.